Thanks for coming tonight. What lights my fire is something that burns inside everything on Earth. It's been poetically called the fire of life by someone you're about to meet, or mundanely bioenergetics or energy metabolism. So I want to tell you how I got interested in it and some of the interesting methods and instruments like this underwater robot that I've developed in order to understand the fire of life. So I need to introduce you to two Maxes and a Tom. The fire of life phrase comes from Max number 1. That's Max Kleiber, who was a Swiss scientist who spent actually most of his career here in this country at the University of California in Davis, working on increasing farm animal production. Max number 2 is in the upper right corner. That's Max Rubner, a German who spent his career at the University of Marburg. And anybody who snuck in a package of snack food during this talk has Max number 2 to thank for the fact that there's a calorie count on that bar. He's the one that established that a calorie is a calorie is a calorie from a bioenergetic standpoint. The Tom in the picture is Tom McMahon, and he became my mentor at about the age that all of you are now when I was an undergraduate, and later was one of my co-advisors during my PhD. All three of these intellects were interested with a very deceptively simple equation up there, namely that the metabolism of an organism, of a living thing, was proportional to that animal's or plant's size. And that it was describable by a simple power law relationship where we had an exponent A. Max was studying farm animals, Max number 1, and found from studies that he did on things like cows and sheep and pigs that the exponent was close to about 3/4. And about the time I met Tom, he came up with one of the first theories to explain why the scaling relationship pertained. And he reasoned that as animals got bigger, the bones that supported them needed to flex by about the same proportional amount. You didn't want an elephant getting floppy when it became an adult elephant versus when it was a baby. Max Rubner was one of the first, though, to think about this from a first principles, and he thought the exponent should be 2/3. That's the relationship between surface area to volume, and his reasoning went like this. If I'm an endotherm, like a dog, which is what he worked on, and I'm producing heat, that heat has to escape through the skin of the animal. So as the dog gets bigger, it better slow down its metabolic rate because as the volume goes up, the surface area is not increasing at the same rate as the volume. It was Kleiber's rule, though, that ended up in the textbooks. Some of you probably have heard of this rule. It may not have been called Kleiber's rule, but you might have heard of it as the mouse-elephant curve in introductory biology. To make the mouse-elephant curve a little more concrete, here's a cute Labrador retriever. My wife breeds and shows these animals, so these are actually hers. And if that cute puppy grows up to be an adult someday, if that prize-winning adult has a litter, you better lay in more dog food because 25 kilograms of puppies eats a heck of a lot more dog food, I have firsthand experience here, than the adult does. And that's because the fire of life is burning much brighter on a per unit mass basis in the puppies than it is in the adult. Well, about this time, I was an undergraduate. That's me on the left, and my other co-advisor, who was then my work-study employer, is in the middle there, Ken Siebens. And my work-study job was actually diving out at the Marine Science Center, working on all these marvelous organisms that don't look like puppies at all up there in the top row. Those are all invertebrates from different phyla, and they coat the rock walls underwater in ecosystems around the planet. Some of those are sea anemones, soft coral, sea squirts. Tom prompted me to think about the fire of life and how it changed with size in these organisms. And when I went to the literature, I discovered all kinds of apologies. People would study the scaling of respiration in corals or sea anemones, and they would get exponents very different than 0.75 or the 2/3 that Max Rubner thought should pertain. And they would always apologize at the end of the paper in the discussion saying, well, we hope to see 0.75 scaling, but we didn't. We're sorry, we must have had experimental error. And I didn't think that perhaps that was the case. These were all careful scientists. When I was underwater with Ken Siebens studying these organisms, I noticed that all these creatures were immersed in a moving fluid. There's motion in the ocean all the time. And so I started an informal study on my own as an undergraduate of fluid mechanics. I started reading fluid mechanics books. I went to the library and I checked out 16mm movies. There was no YouTube back then. These are some of them that were made more than half a century ago by the National Science Foundation to encourage people your age to go into engineering. They're quite beautiful. And when you watch fluids in motion, you can learn some things qualitatively very quickly. If you look at the bottom two movies, you can see what is a very important phenomenon in nature. It's called a boundary layer. The fluid closer to the surface is moving slower than fluid further away. And if you roughen that boundary, like the movie on the left shows, you get a lot more mixing, and that's due to a phenomenon known as turbulence. If you look at the top two movies, the organism itself can create large disturbances, large coherent structures in the flow called eddies. I started thinking about metabolism, and I realized that everything that goes in and out of these creatures that are stuck in one place with water moving past them has to pass through the boundary layer. The transfer of material in and out of the organism at the boundary was potentially a key clue as to how metabolism might work. So I decided to do my own Kleiber-Rubner type experiments. I actually got to live underwater in these underwater space stations while I was still a graduate student. I've continued doing that ever since. And I decided to bring the laboratory to the bottom of the sea. What we're looking at in the movie are some metabolism chambers, and those brown objects there are actually coral colonies. And what I'm doing is I'm measuring their respiration rate, their fire of life, by blowing water past them at different speeds. And what I found was something stunning, that biomass alone was not enough to explain the exponent, that if you actually measured the flow speed and looked at the shape, you could get a diversity of exponents, that there was no Kleiber's rule constant for these organisms, but it was still predictable from first principles of fluid mechanics and chemical engineering. About this time I was thinking bigger and became fascinated with the notion that all these marvelous creatures that form these marine ecosystems could make structures like the Great Barrier Reef in Australia that you can see from outer space, but that biomass was accumulating from these small scale processes that were happening at the scale of millimeters to centimeters, at the scale of the coral polyp. So I started wondering whether the landscape itself might have a metabolism that we could measure. So I built another instrument that a diver could deploy. And this was a device for actually measuring the flux of oxygen in and out of patches of the seafloor. But you could only do that in one spot. And we're trying to understand dynamics at a large, large scale. So that's where the underwater robot came to life. This is a free-swimming, autonomous underwater vehicle I developed. And with it, you can turn the whole ecosystem into your respirometer if you put the right sensor on board. You can swim these devices over respiring or photosynthesizing landscapes, and you can collect data like this that show you oxygen transactions at the scale of football fields. And with that kind of data, you can now start to build mathematical models of how fast the whole seafloor, which is a collection of all these weird and wonderful creatures we saw a few slides ago, is respiring or photosynthesizing. And the stunning discovery that came out of this, and this is work still in progress that we're expanding to other ecosystems, is that that exponent can actually be larger than anything it can be for a single organism. At least for these organisms it can. And that's because they're able, the landscape is able to take advantage of mixing in the ocean at an oceanic scale that you don't get when you look at the fluid mechanics around a single creature. So I'd like to conclude my little talk here with a dedication to some amazing human beings. I'd like to dedicate this to Max number one, is the first of three people I'd like to dedicate this talk to. Max's work on allometry helped increase food security in this country. By knowing what the energetic needs of a farm animal was as it got bigger, you could avoid wasting food that would go into the feedlot to produce those animals. There was a collapse of farming that coincided with a collapse of the economy during the Great Depression, and both my parents grew up during that time. That's a picture of misery of Florence Thomas there. She's a destitute mother of 7, a pea picker in California. That's a famous picture taken by the documentarian Dorothea Lange. She was employed by the Works Projects Administration. So about the time that Max #1 got interested in allometry, the men of families like Mrs. Thompson there, they were called hobos at the time, and they rode the rails because food was so scarce and jobs were so scarce. And they ended up sometimes here in North Tonawanda, where my grandparents lived. My grandfather was a Danish immigrant. He changed his name when he married my grandma. You can see the rail lines coming in here next to the Niagara River. North Tonawanda was the lumber capital of North America at the time. Niagara Falls is just a few miles down the river there, and some of the hobos would end up in their backyard, and so they would give them some work to do and invite them to the dinner table. So with that, I'll take questions, and thank you for your kind attention tonight.
Thank you very much. Very interesting talk. Questions? So Mark, you explained the intellectual reason why you built robots. But tell me, when you were a kid, did you read robot comics?
Oh yes.
So there might be some other.
In fact, when I moved here last year, we had to pack up the house in Williamsburg, and I found a book that I'd had as a child that was actually funded by the National Science Foundation, and it was how robots were going to change the future. And I was flipping through it, going, a little tour down memory lane. I used to be, obsessed with this book when I was like 8. And all of a sudden my jaw dropped. I got to the end of the book, and there was a picture of a robot in the water studying whales in Antarctica. And what was interesting was the shape of the robot with its fin out of the water is a dead ringer for the robot you saw me build there. The prototype was the one that you saw in the movie. And I don't know if I've subconsciously been carrying that design around for 30 years or so before I actually built the robot, but that was a surprise.
Well, I think we'll wrap up. It's been a wonderful experience. Thank you, Mark.
Thank you.